name is Jennifer Gaddy, and I'm a fifth-year PhD student in the Department of Microbiology. And my research project, deals with working on a bacterium that causes pneumonia in soldiers returning from Iraq, and also wound infections in soldiers returning from Iraq, and immunocompromised people. And so basically what we're looking at are the genes and gene products that are involved in making the bacteria adhere to surfaces such as lung epithelial cells, and also plastics, intubation tubes, patients' charts, things like that. So what we work on is trying to knock out genes that are in the bacterium that are involved in making the bacterium sticky, and then seeing what sort of mutants that we can make that don't have that sticky phenotype. And then we try to sequence the genes that we've interrupted, and that's when we use this facility a lot, is to determine the genes that are found in the bacterium that are involved in this adherence.
My name is Ben Phillip. I'm a third-year PhD student in the zoology department, and I work in Rick Lee's lab. Our lab focuses on how turtles, frogs, and insects tolerate environmental stresses like freezing and desiccation, dehydration. So my focus here in the lab is to look at some proteins that play an important role in that tolerance of the stress.
I'm George Copenhaver. I'm a senior in microbiology, and I'm currently working on a research project considering microbes that are becoming resistant to antibiotic treatment in hospital settings. So what we do is we target their metal uptake system, and because microbes require iron in their metabolism, we administer an antimicrobial metal such as gallium, and the microbes will take up the gallium, but it will be not used in the same way as the iron and cause them to pretty much stop metabolizing and die. In certain conditions, that's very favorable because in environments where iron is limiting, the microbes will prefer the gallium over the iron and end up killing themselves. So it's very interesting, and I'm very excited about the results because it can be used for possibly other microbes for immunocompromised patients that might be infected.
Hi, my name is Andrew Hong. I'm currently working with Dr. Haas in this lab. I'm currently working on the random mutations of the enzyme that we're working on, and we're using robotics in this lab to purify the enzyme. And the whole purpose of doing this lab is to figure out the important function of the enzyme.
Hi, my name is Chika, and I'm a postdoctoral student here at Miami. I study proteins and how proteins change over time. With the treatment that you give to a cell, because most people don't know that DNA on its own cannot do anything unless it's converted to proteins, and it's proteins that end up building a cell. And so I studied how those proteins change over time and how we can learn from what proteins do in a cell.